described as pollen sacs inside the anther. They represent microsporangia. Each of these pollen sac contains a large number of diploid cells, called pollen mother cells. These pollen mother cells undergo meiosis and form 4 haploid cells, which initially remain together, forming a structure called tetrad. These tetrads get released to represent individual pollen grains, and these pollen grains represent the microspores. Each pollen grain, when it is mature, is roughly spherical and has a 2-layered wall. The outer layer is called exine and the inner layer is called intine. Exine and intine. Exine is spiny in outline and it contains the secretion produced by the tapetum, that is sporopollenin. Sporopollenin will be present in the exine. Inside the pollen grain, you will find 2 cells. The haploid cell resulting inside each tetrad divides into 2 mitotically, forming 2 cells. One is a larger cell called generative cell. At the time of its release, the pollen grain has a 2-layered wall, outer exine, inner intine. Exine is spiny in appearance and it contains the chemical sporopollenin. The haploid cell in the pollen grain divides mitotically and produces 2 cells. The larger one is called vegetative cell and the smaller one is called generative cell. The generative cell actually represents the male gametophyte. The vegetative cell is involved in the formation of a pollen tube which helps in pollination. This will be the structure of the pollen grain when it is released.